Hello everyone and welcome to Aloysius Academia. In a new lesson today, we will cover everything there is to know about mitosis. We will go through the different stages. We will understand it in more depth, and everything is going to be specific for A-level biology. So please have a look at the revision booklets in the bio below. These revision booklets will have a keyword bank which will help you to develop your answers and ace your exams. Let's start. So what is mitosis? So mitosis basically is a division of cells that results in two daughter cells. So the word mitosis refers to a specific type of cell division. And what happens specifically in this division is that a parent cell will be dividing in two daughter cells, and these daughter cells will have an exact copy of DNA and the same number of chromosomes as the parent cell. And specific terms that you need to learn for this lesson is what is a parent cell, which is going to be the original cell. For example, in this case, a random skin cell. And then you would have the daughter cells, which are the cells that produced after the mitosis or after cell division. You need to pay close attention to what happens to the DNA because that's what makes mitosis different to meiosis. So in mitosis, as we all know, that in our, for example, skin cells, all of them would have DNA, and our DNA is made up of two strands. Each strand we inherited from one parent. So, for example, one strand from our mother and one strand from our father. And that's very important. We will cover what the DNA is and why is it a double helix in a future video. But for now, just make sure that you understand that the parent cell would have a DNA inside it. So what happens to this DNA before the parent cell divides into the daughter cells? They duplicate their DNA, which means that they would have twice the amount of DNA that they usually have when they are not dividing. Why do they duplicate their DNA? That's because the daughter cells would need to have an exact copy of the DNA. So the duplicated DNA will be split into two different cells. So we will have the one DNA will go to this daughter cell, and the other DNA will be split into the other daughter cell. Okay, so at the end of mitosis, what's going to happen is that we will end up with two cells that are genetically identical, okay, and they are also identical to the original cell that they divided from, okay, except in specific cases where the daughter cells might be different to one another based on what mutations they developed, and we will cover also in a future video everything there's to know about mutations and how they might affect the cell. There's a key point that you need to understand that mitosis is always preceded by a period when the cell is not dividing. Okay, so in that state there, that's the period when the cell is not divided, still hasn't divided into two daughter cells. This state where the cell is just staying as it is, it's called the interphase stage. It's a stage that precedes mitosis, and the cell in that stage is not dividing. We will cover all of these points in more detail in the following slides. So let's cover now the interphase stage. What is interphase? Basically, interphase is not one of mitosis stages, okay? So it happens before mitosis starts, and it is a very essential stage for the development of the cell. So because during interphase, the cell is very, very active, so it has metabolism, so it's not quiescent, they are active. They grow, they make protein, they also carry out lots of normal function that the cell would do. And most importantly, for this lesson, you need to know that during interphase, DNA replication happens. A very important exam point that you need to remember that many students, they might get confused of interphase and mitosis. You need to learn that DNA replication takes place during interphase before mitosis starts. And after DNA replication, those two copies of the DNA that were produced, they will remain joined at a region called centromere. And all that happens in the parent cell before the two cells divide. Also, there is an important information about chromosomes that during interphase, chromosomes are invisible. What do we mean with that? So during interphase, you would have a cell like that, and within the nucleus, the DNA is still there. However, it's not tightly collided or condensed. We will cover later on what is the shape of the chromosome. But you need to learn now at this stage that during interphase, the DNA is in a more spread out form, and this spread out form is called chromatin. That's very important keyword to remember. So that's chromatin. In this form, the DNA is referred to as a chromatin. And in general, you cannot see distinct chromosome during interphase. So if you have a bunch of cells that you're looking at them under the microscope, and you see that the nucleus has this the form of DNA, but they do not have the distinct shape of chromosomes, then you would know that this cell is going through interphase. So now let's have a closer look at chromatin and chromosomes, and let's understand these two terms, because sometimes in A-level students might confuse these two terms together. So when we say chromatin, we are basically referring to the loose DNA, to a DNA that is in a loose state and uncoiled state, exactly as we see it in this image here. So it's just a term used to refer to how tightly packed the DNA is. So chromatin, DNA is loose and coiled. What happens during interphase? The benefit of the fact that the DNA is loose, this allows protein in the cell to access the DNA and be able to read for protein production, and that's we call it transcription. We covered it in a different video, and also will allow the proteins and enzymes to access the DNA during DNA replication and allow the cell to replicate its DNA. Because if the DNA is tightly condensed, then the protein wouldn't be able to access specific regions in the DNA, and that would prevent protein production and also prevent DNA replication. That's why it's essential during interphase for the DNA to be loose to allow protein and enzymes to access the DNA. The other form of DNA is called chromosomes. So in chromosomes, the DNA, as we can see in this image, they are condensed and tightly coiled. Each of these are chromosomes. They are DNA that got condensed and tightly coiled, and this can be seen during mitosis. And that's a big distinction between interphase and mitosis. If you look at cells under the microscope, if you see these distinctive structures inside the nucleus of the cell, this would mean that the cell is in mitosis. However, if the DNA is loose, this means that the cell is in interphase. Why it's important for the cells to have this chromosomes during mitosis? Because this will allow the spread of the DNA between the two daughter cells more accurate, and prevent any mistakes. It's going to be much more difficult to split the DNA into two daughter cells if it is like that in a loose form. So just generally, chromatin and chromosomes are made of the same DNA. They are just terms used to refer to how tightly the DNA is packed. And now let's have a look at a different term. So now, for now, we understood the difference between chromatin and chromosomes. Now let's have a look at chromatid with a D at the end. So as we said previously, chromosomes are basically a structure of DNA, carries genetic materials, and it's only visible during mitosis, and the chromosomes, they are condensed form of the DNA. So they are not a loose form of the DNA, they are very condensed, coiled form of the DNA. That's called a chromosome, one chromosome. So what happens, then what chromatid are referring to? So chromatid are referring to a different thing. As we said during interphase, the DNA will be replicated. So if we have this chromosome here, which is made up of one thread of DNA, and when we say a thread, it's just a DNA of a double helix of a DNA, and it's compacted into one chromosome. So what happens is that this one compacted DNA will be duplicated into an identical copy. So these two identical copies of the chromosome, of this chromosome, they are called sister chromatid. So basically, chromatids are one of the two identical copies of a replicated chromosome. That's one chromatid and that's one chromatid. So before the DNA replication, we call it chromosomes. This one, it's called chromosome. after the chromosome replicated itself, now it's called chromatid. So that's one chromatid and that's one chromatid. Together are sister chromatid. And that's only true after DNA replication. And as we said here, the two chromatids are joined together at what is known the centromere. So centromere is the region which join two chromatid, which means two identical chromosomes together. So things that you need to remember that before DNA replication, during interphase, the DNA is the chromosomes are not visible as distinct chromosomes. So you'll just see a DNA, and in that form, it's called ChromatiN with an N at the end. However, after DNA replication, which is still interphase, and before mitosis, the DNA will be still invisible and we cannot see the distinctive structures of the chromosome. However, at that point, the DNA will have had duplicated, and then each DNA will have two identical copies, and then they will get condensed to chromosomes and then bind together by a centromere, making two sister chromatids. So now we understand all that. Let's have a look at the different stages of mitosis. So basically mitosis is a continuous process, but just for convenience and to make it easier to understand, usually in textbooks, you will find it split into four main stages. So the stages are prophase, metaphase, anaphase, and telophase. If you focus, you will see that there's no interphase here. And as we said, because interphase is something that happens before mitosis. So at this stage in the interphase, the cell is not dividing and is going to be in this shape. And then in prophase, which is the first stage of mitosis, we can see that the chromosomes start becoming distinctive and visible inside the nucleus. The other two are metaphase and anaphase. We will cover each of these stages in more depth in the coming slides, but for now, just have a look in terms of the steps. So the next stage after prophase is metaphase, where chromosomes will be lined at the middle of the cell. And then you will have anaphase, where the chromosomes, our sister chromatid, will be split into different sides of the cell. And lastly, in the telophase, the nucleus will reform, the DNA is splitted, and then what happens is that the cytoplasm will start splitting. The whole process of dividing the cytoplasm and the cell membrane is called the cytokinesis. So as we can see the whole process, we started with a parent cell. In the interphase, we ended up with two daughter cells, and cytokinesis is not part of mitosis, but it is a final stage where the cytoplasm and the cell membrane divides. But mainly, these are the four main stages of mitosis. So let's zoom in now into prophase, which has several important things that happens into it. And as we said before, the first thing is that chromosomes will become visible in prophase. So what happens is that they turn from being a long thin threads of DNA that are uncoiled and loose, they will become shortened, more thickened and condensed and tightly packed. And then this give you this distinctive structures. What happens in animal cells in specific, because what happens in animal cells and plant cells are different, and we will cover both of them. But in animal cells specifically, there are two organelles. They are called centrioles. So these two organelles. What happens is that in prophase, the centrioles will move to opposite ends of the cells, inside the cell, and in the exam you might find this referred to as the poles of the cell. So you need to remember that as a keyword, that centrioles will move to opposite poles in the cell during prophase. From each centriole, spindle fibers develop and stretch from one pole to the other pole. All spindle fibers together are called the spindle apparatus. And that's basically what happens in prophase during mitosis in animal cells. So prophase in plant cell, it's a bit different to animal cells, and that's because plant cells do not have centrioles. But they still have the spindle apparatus, as we can see in this image there. This indicates that centrioles are not essential for making spindle fibers, because in plant cell, as we can see, spindle fibers are there, but there's no centrioles. And that again happens during prophase. Other stuff that are important, which happen during interphase, is the disappearance of nucleolus. So what is nucleolus? Nucleolus is basically a dense region inside the nucleus that helps with making ribosomes. We cover in a different video. I think when we do mRNA translation, we will understand more the function of ribosomes. But you need to understand that nucleolus will disappear during prophase. Also, the nuclear envelope, as we can see here, will break down. The breakdown of nuclear envelope, which that's true for animal and plant cells. Once the nuclear envelope is broken down, the chromosomes and the DNA will be left free in the cytoplasm. And what happens then is that the spindle fibers will start binding to the centromere of the chromosomes and start drawing the chromosomes toward the middle or the equator of the cell, which now we will see in the metaphase. So in metaphase, specific things also happens that are different to prophase. Again, as we said, chromosomes will be clearly visible, as we can see here, clearly visible during metaphase, and each chromosome will be made up of two chromatids. As we mentioned at the beginning, that's going to be one chromatid, and that's one chromatid. They are two sister chromatids. And what's unique about these chromatids, that each chromatid is an identical copy of the DNA. So this is one chromatid, and then that's the identical copy of the other chromatid. Is the same true for this sister chromatid, and this sister chromatid, and this sister chromatid. So each sister chromatids are identical copies of the DNA. Okay. Then the chromatids are joined together by the centromere. As we can see, each sister chromatid are joined together by centromeres. And then the microtubules, or what we call spindle fibers, so you might find them in the exam named as microtubules. So they comes from the poles and they attach to the centromere of the sister chromatid. Then the sister chromatids will be pulled and arranged at the equator of the cell. When we say the equator of the cell, we just mean the center of the cell. That's very important. So for exam wording and keywords, you need to say it and write it like that. So you would say that chromosomes line up at the equator of the cell in metaphase. So once the chromosomes line up at the equator of the cell, now the next stage starts, which is the anaphase. During the anaphase, different things happens. The first thing is that the centromeres that used to join two sister chromatid together, it will be divided into two pieces. And then the spindle fibers, as we can see here, these spindle fibers will be contracting, pulling the sister chromatid toward opposite poles. So these sister chromatid, the centromere will break down or divides, and then the spindle fibers, this one will contract and pulls this sister chromatid toward this side, and this fiber will contract and pull this sister chromatid into this side. The chromatids will move rapidly to the opposite pole inside the cell. At this very moment, each chromatid, once the chromatids are separated, now we will refer to each one as a chromosome. So because the sister chromatid has now been separated, so that's one chromosome and that's one chromosome. And as it's very important to remember that the spindles will contract and this will pull the sister chromatid apart, and then each one will go to a different pole. You need to remember that the energy provided, or the energy required for the spindle fibers to contract, is provided by the mitochondria. So mitochondria is an organelle inside the cell, and it provides energy. So what happens is that you will find mitochondria gathered around these spindle fibers to be ready to provide energy for the spindle fibers to contract and then pull the chromosomes apart. If the cells are treated with chemicals that destroy the spindles, this will what happens, the sister chromatids will remain in the equator of the cell, and the cells will not be pulled or moved to opposite poles. You will see in the next stage why pulling the chromosomes to different poles is very important to finalize mitosis. So finally now we come to the last stage of mitosis, which is named telophase. During telophase, also important points happens. The first thing that happens is that chromosomes reach different poles, as we showed at the end of the anaphase. Different chromosomes went to a different poles. Once the chromosomes went to different poles, the chromosomes will become again longer and thinner, so they are becoming chromatin again, because in that form the DNA is called chromatin. However, when they are condensed, they are called chromosome. So in anaphase, chromosomes will turn back into chromatin, become long and thin. Once that happens, the spindle fibers will disintegrate. So the spindle fibers that used to go across the cell in the previous stage, now they disintegrate. The nuclear envelope will start reforming, forming the nucleus, also along with the nucleolus. So all these happen in the telophase. Finally, what happens is that the cytoplasm with the cell membrane, they will divide, producing two daughter cells at the end of the process. And the process of dividing the cytoplasm is called cytokinesis. So cytokinesis is not a stage on its own, but it's rather a process of dividing the cytoplasm. So collectively, during telophase, the nucleus is restored, and cytokinesis finishes the cell division by dividing the cytoplasm. So in a summary of the full cycle or the full stages of mitosis, we will start with interphase, which is where cell actively synthesizing protein, chromosomes are not visible, and DNA replicates before mitosis. So interphase is not a stage of mitosis, it happens before mitosis. The first stage of mitosis is prophase. The main things that happens during prophase: number one, chromosomes condense and become visible, nuclear envelope breaks down, nucleolus disappear, and spindle fibers form. Then it comes metaphase, which is mainly chromosomes line up at the equator. Then you have the anaphase. Chromatids are separated and moved and pulled to different poles by the spindle fibers. And then we would have telophase, where nuclear envelope reform, chromosomes become chromatin again, which means the thin thread that are not condensed, and then spindle fibers will disappear again. Cytokinesis finally divides the cytoplasm, producing two daughter cells. Please have a look at the revision booklets in the bio below. It will help you a lot. It's very valuable. It provides a very valuable keyword bank and all the things that have been assessed on this topic for the past 12 years. So have a look at them, and please like, subscribe, and comment, and thank you for watching. See you in the next lesson.